Right. Hello, everyone. Welcome to the MIF++ seminar this year 2025. It's a great pleasure to introduce Professor Cesar Ramirez-Sarmiento from Santiago, so different places in Santiago, Chile. And Cesar will talk about metamorphic proteins and how to find them. Over to you, Cesar, please.
Great. Thank you so much for the introduction. I know this is kind of a substitute lecture from what you expected because Lauren has some issues. But Lauren and I have been collaborating for a while, and so she suggested my name for giving a presentation about this kind of very rare but not so rare proteins. And so I will tell you a little bit about how they work, how we study them, and how we're trying to push the boundaries in terms of particularly computational stuff to try to understand how they operate. So before I do that, I'll show you the place where I'm sitting at. So I am in Santiago in Chile. This is the STEM building, which is located in the School of Engineering from the Pontifical Catholic University of Chile. And we are located on the 7th floor with a bunch of other folks that work on tissue engineering, optogenetics, quantitative physiology, biofilms, and other plethora of different research fields. So I believe I'm the only one working on protein science. And so the view that you have here is of the Andes during the winter, which is actually taken from this balcony over here. So this is kind of a beautiful place to be at. We have different labs for experiments. So we have 3 different labs for doing experiments with bacteria and also animal cells, and also prototyping of different devices for measuring different observables from experiments that we do in the lab. But also we have access to our own clusters for computational work and different high-end desktop computers for doing computational science. So the talk of today is going to be a mixture of talking about what we do in the computers and then how we validate what we're seeing in the computer with an experiment of a different kind. And I'll be brief in explaining the experiment or the idea of the experiment, and I will focus more on the computational part of things. All right, so we work on 3 different things in the lab. So we work on the emergence of novel protein structures in nature. So we know that there's a bunch of them, and we know that protein structure dictates to a large extent function. So we have myriad protein structures that explain how proteins bind to different genes to express these genes, or they bind to different molecules to do chemical reactions, which are enzymes. We also know that they can be on the membrane, allowing the transport of different ions. But then we don't know so much about how we went from one structure to another, whether there's connections that explain how the diversification of different protein folds actually happen. We don't have a clue about that. And so what we study in the lab and what other people work in the field internationally is about these so-called metamorphic proteins or fold switching proteins. And so the cool thing about these proteins is that they have not one structure, which is the typical paradigm of proteins, is that if you have one amino acid sequence, then you have one structure that explains one function. What we have here is actually that one sequence encodes for 2 different structures, now encoding for 2 different functions or for a regulation of a function. And so they're kind of very important. And we will pretty much talk just about that for the remainder of this presentation. And so, but for the people that are interested in other topics, we also work on plastic degrading enzymes, so understanding the emergence of novel protein functions, and also on some biomedical design, which is in this case the production of photosynthetic blood using photosynthetic organisms modified with proteins for this purpose. But okay, so why are we so intrigued by fold switching proteins, or why do we believe that they are kind of a way of seeing of how different protein structures evolve in nature? Because proteins are dynamic. We typically see pictures about proteins and protein structures, and these structures are very beautiful. They are represented in cartoon representations with helices in these cylindrical twisted shapes, and with beta sheets like arrows, so pretty familiar for most folks working on protein structures to recognize different patterns of secondary structure representation. But we always see pictures, we never see dynamics, but proteins are dynamic. And an example is the production of ATP. So this is made by an enzyme that is called ATP synthase. This is made by all our cells in mitochondria. And what it happens is that we need these movements, these rotations that are allowed by pumping protons in order to allow for the production of ATP. So we need movement. In this case, we need the rotation of this motor in order to produce sufficient energy to pass it to a molecule that will store this energy somehow. And then we have many other examples of protein being dynamic, not so complicated as this one, but all of them are dynamic. So we have, for example, the motions in enzymes for catalyzing reactions. This is adenylate kinase, which produces AMP, so another phosphorylated substrate. And what happens with this enzyme is that it has to undergo these motions to put two substrates together so that the reaction happens. And so again, motions are relevant for function. And then the extreme case of needing motions for protein function will be the intrinsically disordered proteins, in which you have an heterogeneous number of ensembles, not a single structure. And sometimes this collection of ensembles prefers one local minima because they bind to some factor or they bind to some molecule or another protein. But typically in solution, they fluctuate quite a lot. So we have all of these examples. So where do metamorphic proteins or fold switching proteins fit? They fit in a different classification of proteins because they are quite unique in terms of their properties. So the first property that they have is that, as I explained before, we have one single amino acid sequence, which is represented here by this green trace that encodes two different structures. And these two different structures are also dynamic. So you have one helical structure over here, you have one beta structure over here. They are colored in blue because they are the same region in the amino acid sequence, but just two different structures encoded by this single sequence. And the arrows represent here that these are reversible changes. So the protein, once it's encoded in this structure, it can actually change into the other structure and then go back and forth as many times as required for biological function. Each structure, as I said before, encodes a different biological function. And then we have a list of things that are also relevant for these proteins. So first, we know that for all of the 100 metamorphic proteins that we know so far that exist in nature based on experimental results, they are critical to switch or regulate biological functions. If it doesn't happen, then the biological function is lost. And then there's a consequence for the organism for not having that. And I'll explain one of those with an example. Also, these proteins are within protein families, which is collections of proteins that have similar sequences and therefore similar structures. But these protein families in which they are embedded in are non-metamorphic, which might lead for us to say, oh, if we trace the evolution of these proteins, maybe we can understand where did the second alternative fold for these proteins came from, at which branch of the evolutionary pathway this actually emerged. So again, like a very intriguing question. And the other thing is that they participate in essentially many cellular functions that are crucial for survival in all kingdoms of life. And that's another critical part of having a metamorphic protein. And so these are two examples of a metamorphic protein. What you see in the top is a representation of the secondary structure. So helices over here and beta strands over here. What we're representing here is on top one of the structures that is possible for this protein, which is RfaH. And here's the other structure that is possible for this protein, RfaH again. And so we have one structure in which is folded as an alpha helical hairpin, so two helices represented here and here. But when it switches due to a trigger signal, which is in this case DNA bound to RNA polymerase, so basically transcription machinery, the protein goes from having this structure into having this beta barrel, this beautiful beta barrel over here. So we have a drastic change in structure. And what this protein regulates by having this change is the expression of genes that are related to bacteria being infectious, of having the capability of, once they are colonizing, for example, the intestinal tract, then to trigger an infection. So all of the genes that are in charge of that infection are upregulated by this action, by the switching of folds. And in the case of KIB, which is another very intriguing protein, this protein is found in cyanobacteria. It also has this change in which in one condition you have helices and one strand, and in the other condition you have all of those secondary structures switch to the opposite. This protein regulates the circadian cycle of cyanobacteria, and it clicks its timing at 24 hours. So that whenever you have a mutation that breaks this transformation, this switch, the clock is lost, and then there's no ticking. And so these are very intriguing proteins. And we have this analysis from bioinformatics that estimates that actually these are not rare proteins, but actually pretty abundant, and that maybe 4% of the proteins that we know from the protein data bank, which is where we deposit our protein structures, are actually metamorphic. Right. So I'm not going to go into details about how RfaH works, because I mentioned how it works before. It activates expression on genes, but I will talk more about how to analyze these proteins. And so the first questions that we're going to see in the following slides, it's how do metamorphic proteins fold switch? Which is, again, an intriguing question. What is the sequence of events that happens for the protein to go from one state to another, and whether we can actually capture that somehow? And how is this information encoded in their amino acid sequence? And then we will give a hint about what Lauren has been doing in terms of understanding that in more depth. And so motions in proteins are very hard to capture. You can do molecular dynamics, which is a very typical way of looking at motions in proteins. But then the issue we have with molecular dynamics is that they cannot explore very extensive timescales in terms of biological timescales because of the time step that we use for these molecular dynamic simulations. So typically, for a molecular dynamic simulation in which you're using an empirical force field that describes all of the Coulombic interactions and all of the van der Waals and all of those types of interactions, you have some limiting factors for your time step, which is typically capturing the vibrations of different bonds in your structure, such that you will have to run for infinite time a simulation in order to capture a process that it's so complicated as the refolding of a protein from one structure to another. And so what can we do instead? We can replace these empirical force fields, which have a lot of components that explain these interactions between residues in the protein sequence. And we can replace them by contact-based interactions, in which what we will do is that we will take pretty much a ruler, we will take one structure, and we will measure the distances between all of the different residues that we have in our protein. So if our protein has, in this example, 100 residues, we will measure all of the distances between all 100 residues one by one, or well, for each residue pair. And we will have something like this, which is a map of the distances of all residues in our protein. So we have on the X and the Y axis, the residue index of our protein. So our protein goes from 1 to 100. And then we will have different intensities representing that some residues are, for example, in this case, residue 30 with residue 10 are very separated in sequence, but they are together in three-dimensional space. And so we will have these maps of interactions in which we have several residues that are very separated in sequence, but that they are very close together in three-dimensional space. And
Cesar,
so, yeah.
may I ask if these distances are measured between specific atoms, for example, alpha carbon atoms in residues?
Yeah, that's a good question. So when we're measuring this, we're measuring for any atom in the residue that is within certain distance. So we're not capturing, representing this map, we're calculating from the alpha carbons, but in the more general way of setting up the simulations, we are saying we will consider that two residues are in contact if any of their atoms are within a certain distance, whatever that atom might be.
And what's your usual threshold for this distance?
That's a great question too. So typically it's 6 angstroms. So we do filter after we measure all of these distances for a certain distance cutoff. and also for a separation in sequence. And the reason that we say we will consider residues that are separated in sequence by more than 3 residues is because the way that we will represent secondary structure is already encoding that local interaction. Because we will tell the simulations not only, oh, we have these sets of contacts in 3-dimensional space, but we will give some information with an energy function that says, these are the angles in which these residues have to be rotated, or these are the torsions, the dihedral torsions of these residues. So if we added the information about contacts that are too close to each other, like one residue together with the other, that's already considered by the covalent bond, so we don't need that. If we consider that with the second residue, so the residue after the one that it's together, that's considered by the angle. And if we consider the other one, that's considered by the torsions in their secondary structure. So what we're playing around with here is all of the non-bonded interactions. But the bonded interactions are taking care of contiguous segments of protein structure, if that makes sense.
Yes, thank you, Cesar.
Awesome. So when we simplify this, and you have 2 different flavors, I should say, you have a flavor in which you do residue pair interactions, which will be a coarse grain model in which each residue is represented as a bead centered at the alpha carbon. That's one option. You have the other option, which is the one that we have used the most in the last few years, which is all atoms. So all heavy atoms, except for no hydrogens, but all heavy atoms represented, such that we're representing atom-atom pairing interactions and not residue-based any longer. You have these 2 different flavors, but the 2 different flavors work pretty much the same. You have this information about this residue or atom-based contact maps, so which residues or which atoms are interacting and at which distance, and you inform this into some form of energy function that allows you to have an energy minima centered at the distance that you measure with your ruler on your structure. So if that distance is, for example, 1.12 angstroms, for saying some example, then you will use you can use Gaussian potentials, or you can use Lennard-Jones potentials for saying, okay, my minima for that interaction is centered at 1.12. And then whenever I increase the temperature, what will happen is that these residues will move apart, and so interaction is broken. Whenever I lower the temperature, these residues will tend to come together. And if they come too close, then there will be a repulsive barrier that avoids them from overlapping in the simulation. So they will be at the energy minimum. So that's the idea. It's to do temperature changes in your simulation. So you increase the temperature or you lower the temperature for doing the simulations. And you will have something like this, which is like a very simplified method for exploring protein folding. So now the simulations are way faster because you're not restricted by the restrictions that are imposed by empirical force fields in terms of vibrations of covalent bonds. There's estimations that these simulations have the advantage of exploring biological phenomena or biological timescales at 4 orders of magnitude higher than typical force fields. So that's the estimation based on different analysis. And you get some traces like this, which is that if you find the temperature for melting of your protein, so you scan at different temperatures and you find one temperature in which you have melting, you will see these reversible changes in structure. So you will have your protein sitting Q represents the number of native contacts in this contact map. So 1 means all contacts being formed. 0 means no contacts. So this is an indicator of folding. So the higher the value means that the protein is folded. And then sometimes during that temperature that you're simulating at, your protein will unfold and then refold many, many times during the simulation. So this is great for single folders, so proteins that only have one structure. The question is, what can we do for studying now proteins that have 2 structures? And the answer will be pretty simple, which is we combine 2 different contact maps. So pretty straightforward. And I'll explain why we can do that. So you take one structure that has one basin. You take another structure that has another basin. which have a different set of native contacts, which is the important part. The residues that are being forming different contacts in the protein are not the same. And so you can easily actually combine them together to explore their switching their simulations. And these are examples. So these are 3 different proteins that are metamorphic. So we have RfaH, we have a chemokine that is very important for immunity in humans. And we have the circadian cycle protein from cyanobacteria. And what is represented here in dots are the contact maps for each of the states of these proteins. So we have in red one state and in blue the other state. Whatever is in gray, so you can see some small dots that are in gray, those correspond to contacts that are formed by common residues between the 2 different folds. So if residue 60 and 65 are making a contact in this guy, the same residues are making another contact in this guy. And you can see clearly from the map that actually most of the contacts are mutually exclusive. They only represent one state. They do not overlap with the other state. And that allows for simply merging them together into a single energy function for running simulations. So we have done that extensively for proteins like RfaH. This is an example. So this is a simulation in which we start from the alpha helical structure. We see the melting of the tips of the ends, of the helices. And then the protein dissociates. So one dissociates from the other, that it's constant, and the protein switches to the beta barrel. We can run these simulations many, many times. We can see this phenomenon happening many, many times during different simulations, and we can get an energy landscape of that changing structure. And in this case, we found out something quite intriguing, which is that the protein does not completely unfold to get to the other structure. It partially unfolds, but there are some remnants of secondary structure in the protein. And so you have, and here you have a representation of a measurement of the overlapping of the different configurations or ensembles that we explore on the simulation to the native states, so to the true ground states of the protein in the alpha state and the beta state. Here we have that the protein is seated at the alpha state and then switches to the beta states, which are very well represented through a series of intermediates and not through a wholly unfolded protein, which will be sitting over here. We have done the same simulations now with RNA polymerase bound to RfaH, which is even more intriguing because it's about mechanisms of how these proteins exert expression of genes in bacteria. And you can see in this case also the same phenomenon. So the protein goes from the alpha state to the beta state and sits somewhere in the RNA polymerase. These simulations are all atoms. So we have representation of all atoms in the structure. And we can see not only the fluctuations of the protein, but we can capture in the simulations the ordering of, for example, how beta barrel comes together. So we can see that here that some beta strands come together first and some other beta strands come together last across the simulation. And we can run like hundreds of simulations for checking that this is statistically accurate. The overall idea that we have now is that for fold switching proteins like RfaH, we go from one state to the other through a series of intermediates. There are that have some degree of secondary structure of their native states, but some degree of unfoldedness of the protein. And we have reviewed this in like many articles with many other simulations. But one caveat of these simulations that it was typically passed to us when we were going through the review process was, well, is this true? Like, can you actually demonstrate that you have one of these intermediates, that this is actually reality or not just fiction from your merging of contacts? And so we decided to tackle that question by doing a combination of a different type of simulation and experiments. So the different type of simulation was, we want to avoid using a simulation in which we have a path of conformational change. We want to have a simulation in which we can actually extrapolate from just measurements of energy without having to look at the transition between 2 states, which residues are stabilizing one condition and which residues are stabilizing another. And we want to combine that with an experiment that tells us what is the degree of foldedness of each structure? And we want to have that somehow. And so we got this information from Ken Dill, which is a very popular professor working on protein folding, that they developed this method, which is called confine, convert, release. And what it does is that instead of stimulating a transition through a path to understand the free energy difference between two states, what it does is that it eliminates the entropy of the states. So here, this is the confinement part. It poses a potential energy, an external potential energy function that makes that entropy is lost. So you get from an ensemble of states into a microstate. And from that microstate through approximations of harmonic approximations or quasi-harmonic approximations, you can actually get the energy of these microstates. And once you get that energy, you can actually calculate what is the contribution of each residue to the stability of each state. So you have something like this, which is a fully dynamic protein. You exert your external potential energy function that confines your protein into a single microstate. You do the same for the other conformation, and then you can calculate this free energy difference by quasi-harmonic approximation. And by having this, you close this cycle, which is a thermodynamic cycle for getting the energy of your system. So you know now which is the preferred state of your system, and also you can calculate which residue prefers which state. So we did those simulations with experiments. So what was the experiment? The experiment was, how can we look at different parts of the protein while the protein is in the one conformation and another conformation? How can we do that? And so the experiment that we use, it's called hydrogen deuterium exchange mass spectrometry. So proteins typically have hydrogen in their amides, which is represented here by these red spheres. But if you incubate them for certain periods of time, up to 10 minutes, in deuterated buffer, their amides exchange their hydrogens naturally by deuterium. And this only happens if the regions of the protein that are under exchange are solvent exposed. If they are not solvent exposed, then there's no exchange, which is represented here by these guys remaining red, while these other guys, which are like exposed to the solvent, having blue circles now, which represent deuterium. So you can then take your protein, cut it into pieces with a protease, take those pieces and analyze them with a mass spectrometer, and you can get an idea of which regions of the proteins are solvent exposed, which regions of the protein are solvent protected. So how can we use this as a tool for understanding our protein? You can do the experiment for the full-length protein, so the alpha helical state. And if you cut the protein and you just express this domain on its own without the rest of the protein, you have the other state. So you have the two conformations experimentally available. As you do the experiments for these two guys, you will see that they have patterns of differential exposure to deuterium and therefore differential incorporation of deuterium. This is the same peptide for the two different structures in the two different conditions. In the alpha condition, we have very low exposure and therefore very low incorporation of deuterium for one of them. And in the other state, which is the beta barrel, we have a lot more. And so we can say, oh, let's compare the differences in deuterium exchange for different parts of our protein with the differences that we calculated from the simulations, from confining, converting, and releasing our structure. And the patterns are the same. What they explain overall is that the tip of the alpha helical hairpin, which is represented here in red, from both experiments and simulations, is the most stable part of the protein in the alpha helical structure. Whereas the ends of the helices, which are represented in blue in both cases, they melt in order for allowing for the transformation to happen. And this is one of the two intermediates that we saw from our simulations to form during the simulations. So at least we validated that one of these is true. We have done the same analysis for other proteins. So we have done the same for Chi-B. We have been able to demonstrate how Chi-B might fold switch. So in the case of Chi-B, KIB is more complicated because it's an oligomer, so it has more than one polypeptide chain. But if we do the same simulations, we can see the change from being a dimer into a monomer that is not fold switched yet, but then fold switched happens very quickly after the dissociation. We can demonstrate how all of that happens, and then we can validate by experiments that this is actually the case by making mutants that attempt to allow for that fold switch to happen faster. So same set of tools, we can actually take it for understanding what happens with another protein. And there's some other folks that have been working on this with other proteins as well. So this is great. This is our set of strategies for looking at the biophysical part of how metamorphic protein works. But then what we know is that somehow this information has to be encoded in protein sequence. And so we decided to tackle that question by saying, this is well known. What people know is that if you take a collection of protein sequences, you can have two patterns of conservation, let's say. You can have that in one position in your alignment of your multiple sequences that are relative to each other in terms of function and structure, so a protein family. If you take a protein family and you analyze the different patterns of conservation of different residues, you will have two patterns that are dominant. You will have conserved residues, like the one shown here in cyan with a blue circle, that typically are, if we think of an enzyme, it will be a catalytic residue that it's important for catalytic activity. If it's a protein that binds to another protein, it's going to be a residue that it's important for the binding. So they will typically have a functional outcome. And that's why they are so conserved. But then you have other patterns of co-evolving residues, which are represented here in green and yellow, and represented in this structure as red circles. So what happens in this case? Let's say that you have these two red guys forming a contact. What happens if this contact is between two charged residues? If you have a positively charged residue and a negatively charged one, then there's a formation of a salt bridge, which stabilizes that interaction. But if you suddenly change one of the identities of one of these residues, let's say the positively charged, which is here an arginine, by a glutamic acid, then you have negatively charged residue and negatively charged residue. They repel each other, structure is lost, function is lost. So something has to happen if it ever happens that there's a mutation of this residue by another one. There has to be a compensatory mutation on the other side such that the interaction is maintained even after the happening a mutation on this side. And so in the example that I provided, if you have that this residue is positively charged and changes for a negative one, then the compensatory mutation on the other side should be that the negative residue changes for a positively one. And this is represented here. So we have a positive and a negative coming together. And whenever there's a mutation in this position for a negatively charged residue, the compensatory mutation on the other column in the alignment will be a positively charged. So these are co-evolving residues. And we can calculate their frequencies compared to the frequencies of each column independently switching for another identity. And typically what happens is that when we see that there's like a very high probability of observing these correlated mutations in these sites, they explain contacts in three-dimensional space. And this is extremely well known for single folder proteins, but nobody ever asked what happens with metamorphic proteins. And so we did. We took, again, our favorite metamorphic protein, RfaH, for which we have thousands of sequences to work with. We analyzed their patterns of coevolutionary analysis, which is the patterns of co-evolving residues in the multiple sequence alignment. We did a few tricks to try to increase, to try to improve these models by adding more information. So at first we worked with just the proteins that were classified in a database of already curated protein sequences, which is called Interpro. It has something about 2,000 sequences of RfaH. We added metagenomic sequences that we search on our own based on sequence conservation patterns. And also we did something that Lauren came with the idea, which is what if we filter then these sequences by patterns of secondary structure? Because some of these sequences we might know from the very beginning that if we predict their secondary structure, some of them will have beta barrel prediction, which means they are just one state. Some of them will be alpha helical, which just means just one state. And some others will have ambiguous signals. They will have a mixture of alpha and beta. And that will be indicating or suggesting that that protein that might be metamorphic. And so when we added the 2000 sequences, we have in here in red indicates how many non-native contacts we have. So we have our ground truth will be our structure. And then what it's in blue, yellow, and green and purple are the contacts that we're calculating for our protein from that ground truth, like how many contacts are consistent with the ground truth. So from Interpro, if we take the collection of 2000 sequences, it's not enough for having good calculations of or estimations of these co-evolving residues. When we add the metagenomic sequences, though, we have a bump in the number of contacts that we're calculating. If we change methods, so this is direct coupling analysis to Gremlin, which is a more sophisticated method, we have an even higher bump in the number of contacts that we correctly predict compared to the ground truth. And then if we filter out the sequences by the secondary structure prediction, which is we lower the number of sequences that we're analyzing, but we're making sure that these are metamorphic proteins, we keep the values pretty high. And now we can analyze a lesser amount of protein sequences, we still have like good analysis. And if we take those contacts and we replace them by our structure-based contacts that we calculated before for simulations, we can actually predict in the computer from sequence information alone, not using any feedback from structures, the alpha state and the beta state of RfaH. So just by looking at sequence information, we can actually predict these two different states. We don't need the structures any longer. So this is a hint for Lauren, or something that she might talk whenever she has time for it. She has expanded this idea by developing what she called ACE, which is a method for maximizing the number of alternative contacts that we predict from sequences. In her very wonderful study, what they do is that they take the multiple sequence alignment and they filter it in different subfamilies. So first they calculate the contact probability for the whole MSA, and then they calculate the contact probability for different parts of the MSA. So by grouping these sequences in smaller batches, and once they have that, they can actually see that some new contacts emerge due to this differential analysis of whole sequences versus these small batches. Once they have this contact prediction from these small batches, they superimpose all the predictions into a single contact map, which is a huge mixture of signals. And then they use a clustering algorithm to differentiate those that are just noise from the ones that are actually true signals. And they can actually get an understanding of contact maps that now explains two different folds, so two different structures. And they can do this very efficiently. So this is the original analysis without doing all of this like pruning and filtering and clustering of the sequences with the full pipeline. And you can see that for many protein families, you can see these alternate contacts, which explain a new conformation, and that they are actually only found in proteins that have metamorphic behavior and not in proteins that are non-fold switching proteins that are not metamorphic. So this is a wonderful example that she used for classifying proteins, and she was able to classify, if I remember correctly, 56 out of the 100 metamorphic proteins that we know so far were correctly classified by this method that is based on coevolution. So coevolution is a very powerful tool. All right. So I'll jump into very quickly in the last 5 minutes. I will say something about how we can actually learn something from everything that we have done and then put it into something else, which is, of course, AlphaFold. So AlphaFold has been changing dramatically how we work with protein structures, whether we can predict them or not. But there's still challenges, and the metamorphic proteins are a challenge. So in short, what AlphaFold does is that it takes a protein sequence as an input. It passes it through a database for protein sequences to do a search for homologous protein sequences of different organisms. And this multiple sequence alignment is used for calculating distances between different residues. So pretty much coevolutionary information again. And that coevolutionary information is passed into a structure model that will make an idealized structure or a geometric representation of how the structure will look like with some confidence values afterwards. So the important part for the talk is that we have coevolution here. And so you will expect, given that Lauren has demonstrated with her work, and I demonstrated with our work, that coevolution is important for encoding these two different folds, that since AlphaFold is using the same, then AlphaFold should predict two structures. That would be the ideal thing. So you take this collab fold, which is the cloud version of AlphaFold that all people can use freely in their houses without needing a server. You pass your protein sequence for your favorite metamorphic protein, and then you only have one structure and one structure only. You never get the other one. But if you do the same that we did in the experiment, which is that we can chop the protein and just express the domain that fold switches, and you pass only that trench of sequence into a collab fold or AlphaFold, then you get the other structure. So this is somehow saying that AlphaFold might have some information, but this information is just hidden in all of this intertwining of coevolutionary information and structure information and what Lauren probably is going to talk, which is like structure memorization, that these methods are actually memorize some structures rather than knowing how to predict them from scratch. Okay, so how can we alter these patterns of sequence information so that they can actually allow us for predicting a fold switch? And for that, we relied on the use of a concept that it's from paramagnetic spins, and then it was kind of taken into the field of protein folding, which is frustration, energetic frustration in this case. And I can refer to this as a social example. So if you go to the theater or the cinema and you want to sit in a single row, and you go with many different people that have very different types of relationships. So some of them are partners, some of them are friends, some of them don't like each other so much, and you want to sit them in a single row, then you have to find an optimal sitting position such that things like partners that should be sitting together sharing popcorn do not sit away from each other, right? And so what will happen through evolution, through molecular evolution, or through social evolution in this case, is that these patterns of interactions might not change so much, but you might mutate, you might change one person for another so that you optimize sitting position, right? But you will never get to 100%. You will never get to, oh, I satisfied all my social interactions. There's always going to be some trouble. And with proteins, the same thing happens. With proteins, sometimes you have some interactions which are shown in green over here that are satisfying the native state in terms of their energy, their identity of the amino acids at the location in which they're positioned in the structure, satisfies the native state, satisfies this state, but there's some other interactions that are not consistent or that are conflicting with that native state, which I'm showing in red, which are on the other end of the energy distribution that is shown here. So those red guys represent residues that if you change an amino acid for another, you might have a better interaction. Or if you change the location of the positioning of these amino acids in terms of distance or how buried they are in the protein, they might improve actually the interaction. What is known is that there are many proteins that have this unsatisfied context, this frustrated context. And this accounts for around 10% of the context that we have in natural proteins, okay? So why were we so interested in this idea of energetic frustration? Because for proteins like viral proteins that also undergo dramatic changes in structure, they also have the same change. You have some contacts in red, which are shown here, on the prefusion state, so before infecting a cell, that once infection will happen, which is when the structure changes from like this kind of a compressed syringe into an extended syringe for injecting viral material into the cell, these contacts disappeared, and now they are green, which means that they are not in conflict any longer. So they prefer one structure over another. So we said, oh, maybe we can look at frustration as a way of identifying which residues we have to tweak into AlphaFold so that we can predict a different structure. So like I said before, 10% of the contacts that are frustrated are typically located in patches that are on the surface of the protein. They are also located in the active site of enzymes, and they are located in hinged regions in proteins that have domain motions. But we wanted to look at something even more interesting. We are talking about something that you look protein by protein compared to something that you do in AlphaFold with multiple sequences. So the idea was, how can we move from just one structure to a collection of sequences, such as AlphaFold does? And so in here we have just a representation of that number of sequences that we want to look at simultaneously for analyzing energetic frustration. And so what we do, the very simple exercise is we take one structure, we calculate frustration, and then we map frustration into the primary sequence. So we have a sequence now with 21 identities, so 20 amino acids plus gaps. And we also have gray, green, and red for neutral, minimally, or highly frustrated residues. And then you do the same for a collection of proteins that are similar but not the same. So now you have, for example, 30 different proteins, all of different sequence identities to the parental one, which contain different patterns of conservation of sequence and different patterns of conservation of frustration. So you will have positions like this one over here in which you have that all identities are almost the same and all residues are minimally frustrated, they are all green. But you will also have patterns in which you have all residues are actually the same, but all residues are red, meaning, oh, this is an interaction that we are somehow keeping that is pretty bad in conflict with the structure, but it's conserved. And you will have some other patterns in which you don't have that much conservation, like this column over here, in which you have changes by different letters, but then you still have conservation of frustration. So you will have all of these different patterns of conserved sequence, high frustration or low frustration, not conserved sequence, it's still conservation of frustration. So you can use information content for actually filtering for these two different patterns for conservation of sequence, conservation of frustration, calculate it, and then get an idea of how your protein looks like in terms of that, in terms of evolution of frustration in these proteins. This method is called FrustraEvo. This was developed by Gonzalo Parra in Barcelona. I would highly recommend for you to use it. It's pretty fun to use and to see what the results are about. But we use it for our protein, and this is going to be the last slide. So what we saw in our protein is that when we run FrustraEvo and we calculate these patterns of evolutionary conserved frustration, we see that there's 9 residues on the interface between the two different domains that are minimally frustrated. And what we can run with the frustratometer, which is the tool for measuring frustration, is that we can mutate these residues by all other identities. And what we saw was that if we mutate by some residues, so these dots over here are actually residues, these are letters, we see that if we change for some residue, we have increasing frustration, so they turn all red. And if we change for other residues, we have maintenance of frustration in green, which is no conflict. So we ask ourselves, can we do an exercise with this information with AlphaFold? What if we take these 9 very important residues for the interaction between the two domains, and we do 2 exercises? In one of them, we mutate all 9 residues by 9 different residues, different residue identities, but that they maintain frustration as it is. So we will change a leucine by a valine or by another residue, with the idea that the residue will keep its frustration as minimal as possible. And then we will do another exercise in which we will change all 9 residues by 9 other residues, but that they increase frustration. So now they turn red. And we will see what happens with AlphaFold as a predictor of the effect of these mutations. So when we do the analysis, when we replace the residues by other identities that maintain frustration the same, the protein is predicted as an alpha helical structure. There's no fault switch. But if we change the residue identities by highly frustrated mutations, all of the models predict a fault switch. So by tweaking different residues based on frustration analysis, we're actually able to capture which residues are important for the fault switch of the protein. Now, what we were doing was playing around with 9 residues. So we said, oh, this is a lot of residues. Can we do it with hopefully just one, right? And so we came with this idea of comparing our metamorphic protein with a non-metamorphic one and say, all right, of the 9 residues, which one is the different one? So of the 9 residues between non-metamorphic NASG and metamorphic RfaH, 6 residues change identity. And of those six residues, they change their identity. 4 of them change for something that maintains frustration the same. 1 mutation changes it for neutral, which is nothing. But there's only 1 that changes frustration from being minimally frustrated to highly frustrated, which is losing 142 by serine. And when we change with this residue and only this residue in AlphaFold predicts a fault switch. And so all of these things are indicating that in the end, the combination of physics-based approaches with the approaches on AlphaFold based on deep learning are going to be the way of finding which are the hotspots for predicting the residues that are important for a fault switching proteins. Not only that, but we also tried all of the 115 different other residue differences between SG and RfaH, and this is the only one that predicts the fault switch. So this is like very precise. And with that, I think that's enough for me. I have a whole bunch of other information to say, but I'll just go to the acknowledgements. So the work with RfaH in experiments we have done with Irina Artsimovich and Elizabeth Comives in the US. The work on frustration we have done with a bunch of people, both in Barcelona and also in Buenos Aires. So Alfonso Valencia, Gonzalo Parra, María Inés Freiberger, and Diego Ferreiro. And some tweaks that we're doing with AlphaFold for making these modifications that allow for prediction, we have done with Jens Meiler and Philipp Engelberger in Leipzig University. And we are funded by different grants from our National Institute of Research and Development, and also some international grants from Italy and the US. I'm also a member of the Global Investigator Network, so that's been very helpful for doing our research. And with that, open for questions and also for a quick announcement, if I'm allowed. We will have, we had in 2023 our first Latin American workshop on artificial intelligence for protein design in Patagonia, in Puerto Varas. So you cannot see here, but there's a volcano behind these guys. Anyways, we had 60 people attending, and we will have a new one this year in November. So information is going to be out soon. If you're willing to come to beautiful Chile for checking on the volcanoes and then learning about artificial intelligence, you're more than welcome to apply for it. Thank you so much.
Yeah, thank you very much, Cesar. So please thank Cesar, physically or virtually. Yes, and yeah, we have a few minutes for questions. So please, questions for Cesar.
Yeah, if you have any. Otherwise, the other thing that I can do is that I can leave my email in the chat if you want to have a conversation afterwards. But
yes, yeah, feel free to leave the email. I'll stop the recording.